Hi guys, my name is Jeremy T. Policarpio and I am going to discuss my part about the cardiac muscles. What are cardiac muscles? So the muscles are derived from mesoderm, the middle germ layer. So the exception are the sphincter and the dilator muscle of the iris and the myofibrillar cells of the sweat and the mammary glands, which are derived from ectoderm. So the embryonic mesoderm that differentiates into muscle tissues includes the dorsal mesoderm, head mesenchyme, and intermediate mesoderm. So the lateral mesoderm, so which we can see in the figure 12. So the dorsal mesoderm that condenses into bilateral columns adjacent to the neural tube forms the segmentally arranged myotomes. So most intrinsic voluntary muscles of the neck and trunk are differentiated from these myotomes. So some voluntary head musculature or the muscles of eye and the tongue and the limb musculature are derived from myotomes and the lower vertebrates. So the limb muscles in higher vertebrates are mainly derived from lateral plate mesoderm. So in tetrapods or the myoblast component of all truncal of and appendicular muscles originate from summits or the somatic origin. So the voluntary muscles of the bronchial or visceral arches of the head and the neck are derived directly from head mesenchyme. So next, so we can see in here in figure 12 that the differentiation of monomers and the establishment of the primordia of virus muscle groups. So we can see in letter A the early separation of the monomer into an epimer, so that's supplied by the dorsal ramus of the spinal nerve of the level and a hypomer, so supplied by the ventral ramus of the nerve. So in letter B, we can see that the later stage in the differentiation of muscles primodia at abdominal levels. In letter C, the primordial muscles masses at the level of the arm buds. So let's move on to the cardiac muscle. So the cardiac muscle is derived from the splanchnic mesoderm. So the involuntary musculature is differentiated from the intermediate mesoderm, the mesomer or orogenital mesoderm or neoprotomic mesoderm. And the lateral mesoderm is the splanchnic mesoderm and hypomer. So the intermediate mesoderm differentiates into much of the orogenital system. And the lateral mesoderm differentiate into the vascular, digestive, and respiratory system and related structures. So let's move on to the differentiation of striated muscles. So the striated muscles differentiate from the myotomes of the summits and from mesenchyme of the non-meiotic origin. So muscle cells, facial cells, tendon cells, and aponeurotic cells are derived from this structure. So, the pre-muscle cells or the myoblasts migrate as the organism develops. So, when the myotome is adjacent to the neural tube, it receives its initial innervation, which is retained during the subsequent migration of the developing muscles. So, the innervation of the primordial masses and the retention of this innervation during development are significant as means of determining the homology of muscles of different species. So the nerves are probably not concerned with organizing these muscle masses because muscle will develop without any innervation in certain monsters. So the axial musculator. So in vertebrates, the muscles of the neck, the trunk, and tail are derived from myotomes although claims have been made that some myoblasts differentiate endermatomes. So the myotomes develop and migrate laterally and eventually as the epimer to form the dorsal or epixal muscle mass. So the dorsal and the lateral back muscles, and the hypermore to form the ventral and hypixal muscle mass. So the lateral and ventral muscles. So let's move on to the fate of myotomes. So the fate of myotomes during their differentiation into the axial muscles differs into by various vertebrates. So in figure 13, we can see the different muscles, different axial muscles in different vertebrates. So in fishes, the embryonic pattern is retained because the aphyxial and hyphaxial muscles are relatively equal in size. So in the amphibian, two adult patterns are developed in aquatic amphibians such as Necturus. So the embryonic segmental pattern is retained. So in terrestrial amphibians also, such as the frog, the meiotic derivation are modified during the metamorphosis by migration, fusion, and splitting. So as a result, the embryonic pattern is modified as a functional adaptation to life on land. So in other hand, terrestrial animals, reptiles, and mammals, the embryonic meiotic segmentation is altered further than in the terrestrial amphibians. So the epiphyseal muscle form the erector spina back muscle. So some hypophyseal muscle retain vestige of the segmental patterns or the intercostal muscles of abdomen, whereas other muscles are the products of migration, fusion, and splitting during development. Flat abdominal muscles. So the muscle pattern are altered more drastically in birds. So we can see in here in figure 13 the development of muscle from the visceral arches and the myotomes in various vertebrates. So the basic areas in the embryo from which voluntary muscles develop. So in shark, neurons, frog. So in in B, the shark. C, the Necturus, and D is the frog, and cat, and the I is the goose. So this picture is the different of about the myotomes. So that's all my report. Thank you.